Unlocking the secrets of the brain. Neuropsychology. Neuropsychology is the study of the function of the brain by investigating the effects of brain damage on mental functions. Here there is a table of different causes of brain injury. For example, cause of damage, vascular. Description: disruption to blood flow to the brain for a number of reasons, including a blockage, stroke, partial blockage, ischemia, or an enlarged artery, aneurysm. The loss of function depends on the severity of the event and the position of the damage. Tumor, also described as neoplasm, a mass of tissue with no physiological function that grows and disrupts normal functioning. The tumor may cause vascular problems or may destroy neurons. The severity of the tumor depends on its size, growth rate, and position. Gliomas begin with abnormal glial cells. Meninglomas originate in the meninges surrounding the brain. Metastatic tumors originate elsewhere in the body but invade the bloodstream and so travel to the brain. Degenerative disease, a breakdown of neurological material, may be genetic, as in Huntington's, and influenced by environmental factors. Examples include Alzheimer's, Parkinson's, and Korsakoff's diseases. May be critical in the cortex or subcortical beneath the cortex. Each disorder presents with different behavioral and neurological symptoms. Infectious disease. A virus may result in neurological symptoms. HIV, and AIDS tend to result in subcortical damage. Herpes attacks cortical structures. Trauma. A violent assault on the head, be it with a car or a weapon, can result in trauma to the brain. May be closed or open head, depending on whether the skull remains intact. The resulting damage may or may not be at the point of the impact. The trauma may also result in vascular problems. Epilepsy. A transient loss of consciousness resulting from excessive and often focused electrical activity in the brain. Investigated with EEG. Often triggered by a trauma, this disrupts activity across a wide area of the brain and so is hard to investigate cognitively. Neuropsychological tests. Neuropsychologists use a variety of neuropsychological tests to measure verbal and nonverbal behaviors of people who may have suffered brain damage through accident or disease, or even just the aging process. As well as providing diagnostic tools, these tests are also important research tools. Neuropsychology. Single and double dissociation. By studying patterns of impairment and sparing of function on neuropsychological tests across brain-injured patients, neuropsychologists can draw inferences about how brain functions are related to one another in the brain. An important concept here is that of dissociation of function. Put very simply, dissociation is a difference in performance between two tasks. For instance, if a patient or a group of patients with damage to a particular part of the brain performs poorly on verbal dexterity, but well on comprehension, then this provides some evidence that the damaged part of the brain is involved in verbal dexterity, but not comprehension. This kind of evidence is called a single dissociation. Stronger evidence of separation of function comes from double dissociations. In a double dissociation, we see a pattern in which one patient is impaired on task A, but spared on task B, whereas the second patient is impaired on task B, but spared on task A. When we have a double dissociation like this, we can say much more confidently that the differences seen are due to the fact that these two tasks are subversed by functionally separate systems in the brain. Wernicke's aphasia results from damage in the temporal lobe and is primarily manifested as difficulties with speech comprehension. Broca's aphasia results from damage in the frontal lobe and is primarily manifested as difficulties with the production of speech. Lesion studies in animals. Experimental lesion studies with animals are another useful method of learning about brain function. Researchers can produce brain damage or lesions in which specific nervous tissue is destroyed with electricity, with cold or heat, or with chemicals. They can also surgically remove some portion of the brain and study the consequences. Such studies have been particularly useful in constructing animal models of human brain diseases. Electrical recording. Because brain function consists partly of electrical activity, scientists can eavesdrop on the electrical conversations occurring within the brain by measuring that electrical activity. One method is called single-cell recording. It is to measure the activity of single neurons or small groups of neurons by inserting small electrodes in particular areas of the brain. Occasionally, it is possible to do this kind of research in humans, usually when participant is undergoing investigative surgery for epilepsy. Another method, let's see. In addition to measuring the activity of small numbers of neurons, neuroscientists can also measure the activity of much larger groups of neurons. One advantage of this method is that it is possible to measure the responses of large groups of neurons without an invasive procedure. We can record neural activity from the surface of the scalp. Electroencephalography, EEG. Electroencephalography measures the activity of large groups of neurons through a series of electrodes placed on the scalp. Although the EEG is a rather non-specific measure that taps the electrical activity of thousands of neurons in many parts of the brain, specific EEG patterns of temporal fluctuations and firing different oscillation frequencies correspond to certain states of consciousness, such as wakefulness and sleep. This method has been particularly useful for studying neural responses in human infants. Clinicians also use the EEG to detect abnormal electrical patterns that signal the presence of brain disorders. Another recording is called MEG. Magnetoencephalography. Magnetoencephalography, MEG, is a very complex method that makes use of the fact that each time an electrical current is created in a neuron or group of neurons, this generates a magnetic field. MEG records these magnetic fields using an array of highly sensitive magnometers, instruments for measuring very small magnetic fields, placed over the head. The most commonly used magnetometers at the moment are called SQUIDs, superconducting quantum interference devices. MEG is closely related to EEG as it is particularly sensitive to the time course of neural processing. MEG is also better able than EEG to deliver estimates of where in the brain a neural function is taking place and could equally belong with the brain imaging methods described in the next session. Because of its temporal sensitivity, MEG, like EEG, is particularly helpful for informing researchers about the ways in which brain activity oscillates according to particular frequencies of neural firing. MEG is increasingly being introduced into clinical settings to examine potential functional abnormalities in brain activity. Brain imaging, MRI. Magnetic resonance imaging, MRI, creates images based on how atoms in living tissue respond to a magnetic pulse delivered by the device. In magnetic resonance imaging, MRI, a strong magnetic field is passed through the brain and pulsed on and off. The oscillating magnetic field causes the molecules and atoms in the brain or any tissue to respond. When the magnetic field is shut off, the molecules in the brain emit a radio signal, which is then detected and mapped to provide images of the tissue. A more recent method using MRI is DTI, diffusion tensor imaging. Diffusion tensor imaging. DTI measures how water molecules diffuse in tissue. In the brain, water molecules are not free to move in any direction because of the shapes of the cells and structures they inhabit. This means that we can get information about how structures and pathways or tracks in the brain are aligned. This has been particularly useful for providing pictures of how white matter tracks are arrayed in the brain, telling us about how the brain is structurally connected. A particularly important advance in MRI technology is functional MRI, or fMRI. fMRI makes use of the fact that oxygenated and deoxygenated blood respond differently to the magnetic fields used with MRI scans. When there is neural activity, more oxygenated blood is sent to that part of the brain. fMRI researchers thus investigate where the highest concentrations of oxygenated blood are being sent in the brain at any one time and infer on that basis which parts of the brain are most active when a participant is undertaking a particular task. PET, positron emission tomography, PET scans. PET, positron emission tomography, have provided another way of measuring where blood flows to in the brain. Glucose, a natural sugar, is the major nutrient of neurons. So when neurons are active, they consume more glucose. To prepare a patient for a PET scan, a radioactive form of glucose is injected into the bloodstream and travels to the brain, where it circulates in the blood supply. The PET scan measures the energy emitted by the radioactive substance and can thus map out which parts of the brain are the most active. One of the disadvantages of PET involves the injection of radioactive isotopes, which can be harmful if applied frequently. This is one of the reasons why fMRI is a more popular method for imaging brain activity. However, one of the advantages of PET is that it is possible to use radioisotopes to label particular neurotransmitters. This means that PET is still an important method for investigating the role of particular neurotransmitters in brain function. FNIRS, FNIRS, Functional Near Infrared Spectroscopy. Functional Near Infrared Spectroscopy, FNIRS, is a relatively new method for investigating brain function. It involves shining a particular kind of light, near infrared light, into the brain and measuring the ways in which it is reflected back. This method is completely non-invasive as near-infrared light, although we cannot see it, is naturally occurring and able to permeate biological tissue. In fact, near-infrared light is probably shining into your brain right now. Because oxygenated and deoxygenated blood absorb near-infrared light to different extents, it is possible to use FNIRS in a similar way to fMRI, measuring the signatures of blood flow to determine which parts of the brain are the most active. FNIRS is a little limited in comparison to fMRI, as it gives less precise maps of the brain, and it can only really tell us about blood flow to areas close to the scalp. As near infrared illuminates only a few centimeters into the brain. However, it has some important advantages. First, it is a relatively inexpensive technique to use in comparison with fMRI. Second, whereas fMRI simply does not work if the participant moves with FNIRS, it is possible to image the brain of someone who is moving around and acting on their environment. This makes it particularly useful for examining the neural basis of motor behaviors, and also makes it very handy for using with participants who do not usually sit still. Infants, for example. Brain stimulation. An alternative to measuring the activity of neurons is to stimulate them and see what happens as a result. The importance of brain stimulation techniques is that they can show us more directly what brain activity in a certain region or network causes to happen. Transcranial magnetic stimulation, or TMS. Transcranial magnetic stimulation, TMS, is electrical stimulation of a targeted part of the brain via magnetic pulses sent from an electromagnetic coil. Transcranial electrical stimulation, or TES tests, transcranial electrical stimulation tests, is electrical stimulation of the brain by applying a low current to the scalp. The hierarchical brain, structures and behavioral functions. The hindbrain. The hindbrain is the lowest and most primitive level of the brain. As the spinal cord enters the brain, it enlarges to form the structures that compose part of the stalk-like brainstem, which also includes the midbrain. Attached to the hindbrain part of the brainstem is the other major portion of the hindbrain, the cerebellum. The medulla and pons, life support systems. The structures of the brainstem support vital life functions. Included are the medulla and the pons. Medulla plays an important role in vital body functions such as heart rate and respiration. Pons lies just above the medulla and relays sensory information between the cerebral cortex and the cerebellum. Cerebellum, concerned primarily with muscular movement coordination, but also plays a role in learning and memory. Brainstem spans the hindbrain and midbrain. Hindbrain structures in the brainstem include the medulla and the pons. The brainstem supports a number of vital physiological functions. The cerebellum, motor coordination center. The cerebellum is concerned primarily with muscular movement coordination, but also plays a role in learning and memory. The midbrain. Lying just above the hindbrain, the midbrain contains clusters of sensory and motor neurons. The sensory portion of the midbrain contains important relay centers for the visual and auditory systems. The reticular formation, the brain's gatekeeper. Reticular formation acts as a kind of sentry, both alerting higher centers of the brain that messages are coming, and then either blocking those messages or allowing them to go forward. Forebrain, the brain's most recently evolved portion. Cerebrum, the most superior part of the forebrain, comprising the cerebral cortex and several more central structures. Thalamus, sometimes likened to a switchboard that organizes inputs from sensory organs and routes them to the appropriate areas of the brain. The forebrain. The forebrain is the most recently evolved of the brain. Its major structure, the cerebrum, consists of two large hemispheres, a left side and a right side, that wrap around the brainstem as the two halves of a cut grapefruit might wrap around a large spoon. Interesting. The thalamus, the brain's sensory switchboard. The thalamus is located above the midbrain. It resembles two small balls, one within each cerebral hemisphere. The thalamus has sometimes been likened to a switchboard that organizes inputs from sensory organs and routes them to the appropriate areas of the brain. The visual, auditory, and body senses, balance and equilibrium, all have major relay stations in the thalamus. Hypothalamus plays a major role in many aspects of motivation and emotion, including sexual behavior, temperature regulation, sleeping, eating, drinking, and aggression. Limbic system helps coordinate the behaviors needed to satisfy the motivational and emotional urges that arise in the hypothalamus. It is also involved in memory. Hippocampus, involved in forming and retrieving memories. Amygdala, amygdala, underlies emotional behaviors, particularly those linked to aggression and fear. The cerebral cortex, crown of the brain. The cerebral cortex is the most recently evolved part of the forebrain. It's 0.63 centimeters thick sheet consisting primarily of gray matter. This is the outermost layer of the human brain. Motor cortex controls the 600 or more muscles involved in voluntary body movements. Three of these fissures, also known as sulci, are important landmarks. One large fissure runs lengthwise across the top of the brain, dividing it into a right and left hemisphere. This is called the medial longitudinal fissure. Within each hemisphere, the central fissure divides the cerebrum into front or anterior and rear or posterior halves, and a third fissure, the Sylvian fissure, that runs from front to rear along the side of each hemisphere. On the basis of these landmarks, the neurologists have divided each hemisphere into 4 lobes: frontal, parietal, occipital, and temporal. The motor cortex controls the 600 or more muscles involved in voluntary body movements. The sensory cortices, somatosensory cortex, receives sensory input that gives rise to our sensations of heat, touch, and cold, and to our senses of balance and body movement, kinesthesis. Association cortex, involved in many important mental functions, including perception, language, and thought. The frontal lobes constitutes 29% of the cortex. The prefrontal cortex, located just behind the forehead, is the seat of the so-called executive functions.